I did have a question that was asked. I will not take responsibility for this question, but I think I need to ask it anyway. By a show of hands here amongst the group, how many panel members think that long-term hibernation will be feasible for humans during spaceflight?
Oh, I'm in favor.
What's the time frame?
Well, there was no time frame specified, so we'll let the record show that everybody on the panel seemed to think that that was feasible. I think that's kind of an important one to at least ask. So, another question I wanted to throw out here as well is what do you think are the most, and we've talked about this a little bit, but maybe there are others who haven't thrown in their 2 cents, the most promising or even necessary approaches for eventually inducing torpor or hibernation-like states we'll start with in animals other than those who naturally do it. Ultimately humans, obviously, and what those animals might be.
Well, as far as I know, there's been only 3 or 4 ways that this was done. Like, so at least in non-hibernators, there's been, we did the RAFA inhibition, then there is Kelly CAJ approach. Then there was a Russian group with a cocktail, put in a liposome, I think, or in a vesicle with xenon, and there was an antipsychotic, a beta blocker, and magnesium maybe. There were 6 elements, I don't remember them all. And then recently in this Nature paper from Sakurai, using chemogenetics and activating at least part of those Q neurons that they suggest are producing a very strong metabolic suppression. They have done it in rat. It kind of worked. That there are not many data. It's a supplementary figure on the paper, so it's hard to tell exactly how powerful it is. But if we're given a good, as far as I know, there's been 4 ways to simulate somehow torpor always in rodents and rats. The Russian group, I spoke with the first author, and she claimed they've done it in rabbits as well, but I've never seen it published. So that's what I can say.
What about hydrogen sulfide was trendy there for a while, but is everybody in agreement that that's just like a mild poisoning and not a sustainable form of, like, that may drag?
It didn't work in large
sheep.
animals.
Exactly,
Yeah.
it didn't work in sheep, right?
I think it's a chemical hypoxia that's mediated,
mechanism
mediated through adenosine is my guess.
So they hit a Goldilocks zone in the mice there where it worked in terms of not
honestly,
poison them too much. But,
yeah.
Well,
what
if you look at the
with,
paper, in rats, they also use a 17% oxygen atmosphere, so there is also a bit of hypoxia.
And in the mouse study, they regulated their body weight or their body temperature decline with the ambient temperature.
Yeah.
So it was.
Yeah, the original mouse study was also hypoxic. So they used a hypoxic environment, and that is actually ultimately responsible, not the hydrogen sulfide, which nobody's been able to reproduce to my knowledge.
Didn't the sheep people reproduce it in mice, but they couldn't do it in sheep?
Yeah,
mice works.
Hypoxia too.
Yeah.
Okay, we still have 1 more hour, and I really want to get to the questions that we haven't answered. But before we do that, I want to ask 1 question. Is there any difference between male and female in hibernation? Our institute is really trying to take consideration into male versus female astronauts and to be more encompassing. So in this specific field, is there any difference between males?
In ground squirrels, in our studies, the only difference is timing.
Yeah, timing. Yeah,
but
which is
there's
really
absolutely no difference in anything else that we can detect.
Yeah, and
but
the timing,
testosterone,
I think,
testosterone is an enemy of hibernation. So
yeah. So
But
I think the timing, the timing illustrates the neuroendocrine role in regulating the seasonal rhythm.
Mm-hmm.
And that is there's a sex difference there for sure. But the metabolic suppression, the gene expression really is not significantly different. Yeah.
No.
So going back to that testosterone mentioned from Dr. Seri, the muscle recovery after torpor hibernation, is it the same in males versus females, regardless of hormones?
Well, they don't really lose it during the hibernation season, so there's really no recovery at the end in the spring.
Okay.
So in 13-line ground squirrels, there is a little bit of loss in the
Oh.
middle of the hibernation season. It's as if they start using muscle early in the first half of the winter, but they rebuild it in the back half of the winter. And as far as we can tell, there's no difference between males and females. So when they emerge, they're fine.
Okay.
In mice, is harder to push male into torpor than female, and for fast-induced torpor. That's another difference.
Maybe because the females have more fat.
I was thinking about testosterone, so that they don't have the time to prepare. So send female astronauts. That would be my best guess.
All right.
Okay. Dr. Strandman, Dr. Fab, any other questions before we move into the a little bit into the questions from the attendees? I want to make sure that we have time to answer some of those.
Yeah, I have one more question, and this could just be a very big reflection on me and my naivety here. But I know that we've talked a lot about knockouts and trying to understand correlations on understanding, okay, well, here's this finding here, this genetic outcome here, and how we maybe could knock it into another model or knock it in or out or overexpress in the same animal models. And something that I've been wondering, and this goes back to my grad school days, and we looked at knockouts and knock-ins and protein mutations and so forth. But I think about an animal or a tissue or an organ, and as the cell is just doing its day-to-day thing, it's producing these genes and it's producing these proteins, which also impact other proteins, which impact extracellular matrix, which impact cell-to-cell communication, which impact tissue architecture and function, which impact organ. And so I'm wondering, when an animal, when you're trying to modify these genes, would just that one modification be enough to actually change the hibernative state? Because you also have to think about the architecture of the cell with its other networks of communication and that gene expression, and then also cell-cell and extracellular matrix and all of the complexities that follow downstream to that. Has that ever been a concern or a question that you think about?
It's hard to tell. If you use a knockout for a neurotransmitter like NPY and knocking maybe Y1 receptor, then you can show that mice pretty much don't enter torpor. At that point, the only thing you've done is really preventing a neurotransmitter to work on specific neurons. So that's very limited in terms of the activity, the consequences of the gene that was knocked down. If you're going for something that is involving cell metabolism, then I think you're going to find yourself in the more difficult place, because the adaptation of the animal to that knocking will be much wider, and you don't exactly know how the final phenotype is just a result of that gene. That's my vision.
Any other thoughts to that question?
I mean, I think no matter what, you have to look at the whole animal. It's never going to be to recapitulate all of hibernation is never going to be a single target. And honestly, I don't think we'll be able to ever recapitulate the whole thing in humans, because I think there's some species adaptations that is unique in the ground squirrel or in hibernating species to tolerate some of the stresses involved with onset and recovery from torpor. I mean, that's why it's nice to consider, whatever is regulating the global change in gene expression, like epigenetics, or I think epigenetics is more of a potential global influence than microRNA. But
Thank you.
Okay. Okay, thank you for that. Dr. Shrema, sorry. Dr. Sherman, if you want to start reading some of the questions from the public, that would be great.
Sure, I can do that. Note that I'm juggling this live from 100+ questions that have been flowing through. So my apologies if I don't get to everybody's question or I misstate it, so forth. It's the downside of not having an in-person live thing is that it's a lot harder to do interactive discussions and so forth. So bear with me a little bit. But one question that was asked, and it's of particular interest to me, being a brain person, is what is behavioral or cognitive recovery like after induced torpor, and especially, generally speaking, and especially in rats who don't normally do this?
Well, in rats, I don't see any specific consequence after we've been putting in synthetic torpor. I mean, they have this long period of intense sleep, pretty much like regular hibernators do. And the day after, you wouldn't know that was in torpor. But we haven't done any specific memory test or.
I think that's a gap.
Conditioning.
It would be really great to have some resources to do more comprehensive cognitive assessment.
At least on the micro scale, nothing serious, but yeah.
Yeah, that seems like it would be an important one, seeing as we don't want to wake up our astronauts and find out that they can't function.
We've seen in hibernation that they actually learn better 24 hours after they come out. And, there's just this remarkable synaptic plasticity that happens during torpor and arousal that's largely mediated by temperature. And so you can replicate that in a slice by lowering temperature and see these morphologic changes. So it's really interesting to consider what the functional impact. And then, there's a study with the RBM3 RNA binding protein that they used in neurodegenerative models, and they found that it played a role in the synaptic remodeling, and it could substitute for the temperature effect. So rather than lowering temperature and rewarming, they could use the RBM3. And so there's some interesting stuff out there that I think has. And tau, and then reverses. Yep.
Yeah, that is very interesting to know that and be able to track that, and it would be good to fill that gap, I think.
That's forward. A gap.
And some of these questions, since it's just a smattering over the past 4 hours, are going to be a little bit all over the map. But again, bear with me. I tried to clump them reasonably. So from the point of view of space travel, do folks think that warm torpor be less interesting than cold hibernation, where you have reduced oxygen, nutrient consumption, and so forth? Or are there reasons to consider warm hibernation that are in its favor, should we say?
I'm just going like brainstorming here. So I'm be very happy with warm hibernation. My main worry would be, how are you cognitively performing? Because if you're gonna be awake on a spaceship, but let's say drunk, kind of like, that's not, I guess, a good state. So warm would be good.
Have your finger on the controls.
Right. Warm hibernation would be good as long as you're still knocked down. That would be my, or cold would just give you an extra kick in terms of metabolic depression. So definitely you could go deeper if you get colder. You have to weigh the side effect of that and the difficulties. But I think the cognitive part here would play a role. If you're just slowly thinking, that may not be put you in a safe spot on a spaceship.
But warm hibernation would still result in a reduction in resource use, right? Like there is still, metabolic rate is still reduced. It's just not as profound. Like if body temperature, bears go to, I guess, 32 degrees or so, but it's about an 80% reduction in metabolic rate, despite the fact that body temperature is still at. So warm torpor, I guess it depends on how heat is lost over the subject, and how close they assume ambient temperature.
So this
But
question
there'd
is my
still be
own
there'd still be savings. Like there'd still be benefits in terms
Right.
of resources.
So to follow on that, and this is really partially my own ignorance, is there any relationship really between, I mean, there is a relationship between temperature reduction and or portion temperature reduction and the savings on the metabolic side. Is there, formula is a little bit too strong a word, but is there a way to characterize the magnitude of savings that you'll get, or is it just, different species are really different from one another and how that curve looks?
You can calculate it using factoring in Q10, and from experimental data, you can have a guess from the mass and the body of the animal. You can scale it to a certain degree, and you could estimate the amount of energy you could save. For humans, I think it wasn't that much. And if we were a natural hibernator, it would be about 30% saving, I think. I don't want to remember the wrong way. So that's why temperature could give us an extra kick. But then, ambient temperature factors in, and we're a hairless species, so maybe heat loss would be higher. There is some rough way to have an estimate.
And
The
it's mostly based on mass.
But the metabolic rate of bears hibernating, even though they're not so cold, is per kilogram animal the same as the ground squirrel.
Yeah.
Yeah. So that you have to explain other than temperature. So.
Well, yeah, and then if I don't remember wrong the figure, then if you project that, you get the blue whale,
The blue whale,
which per unit of mass would be are in hibernation. Currently.
Yeah.
Which is like a lower limit of, it's been posited as like the lower limit of metabolic rate in a biological organism.
Right.
Yeah.
Right.
That's a really neat idea.
So I think
sorry, Sandy,
one thing that's the jury's still out on is whether or not the cold is protective against either bone or muscle loss. And I think that's a real possibility, that is a component of their resistance to atrophy in both of those systems.
I agree with that.
Definitely interesting. So another question is, has any EEG or other brain activation data during hibernation been related to the molecular findings? Anybody know of any?
Not that I know. We have a lot of EEG recording over there, but I don't think to remember any connection with that. We've done something with orexin knockout lately, and they seems to mice, of course, and they seem to go into torpor exactly like the other do. So, but it would be interesting to go through at least the classic pool of neurotransmitters and the arousal system and see if there is any of those that could favor or prolong eventually torpor. Oxytocin maybe does something like that, I guess, but MPY, but it's not being systematically explored as a further. Kelly, you have any?
No, I was just going to say the only thing, not molecular, but it does really beg the connection between temperature and slow wave sleep, and the role of temperature in regulating slow wave sleep. I think we see that more when we study hibernation than when you study sleep by itself. It really, exaggerates the influence.
Oh, yeah.
So what is the connection? And the reason why I asked with slow wave sleep, and the reason why I ask this is one of the projects that Trish is currently funding involves the auditory stimulation to enhance slow wave sleep in humans. So it's essentially synchronize the auditory bursts with the delta waves to maintain and perhaps deepen or whatever slow wave sleep. And so I'm wondering, since I didn't know about the relationship, what is the relationship between slow wave sleep and hibernation?
Well, my interpretation is that it all begins with metabolic suppression and lowering of body temperature, and that the lowering of body temperature produces the phenomenon of slow wave sleep that then extends into torpor, where you just lower amplitude, you keep losing amplitude and then you're a flat line when you're cold enough. That's my impression. Maybe Matteo or others have.
Yeah, no, we've been more interested in the sleep rebound that you get after turbo hibernation. So you get this huge sleep rebound, and especially a lot of delta waves. And there've been conflicting evidence in the past. And if you deprive the animal from this rebound, some have seen a delay rebound. So this was actual sleep, but some haven't seen that. And so they were suggesting it was just some way the cortex was coming back online. Let's put it that way.
So by rebound, you mean an excess of slow wave delta sleep at the end of a torpor period? Is that it?
Yeah. After our rats, when they come back up at, let's say 35, 34.5, 35, they start sleeping. The AG during synthetic torpor is very, and from what I've seen also during torpor, it's very different from sleep. So someone was suggesting this is a sleep deprivation period too. And has been also proposed that the interbout arousal may have been driven by sleep. So whatever sleep is doing to the body, it can be done during torpor for unknown reason. Then the animal has to wake up, do the sleep, and then go back.
Yeah, but that was proposed by Craig Heller a number of years ago, and he went in and studied it in depth and came to the conclusion that that's not right. So he published a definitive paper saying, no, it's not that they wake up to sleep. So let me just put it out there that we can say that they wake up to sleep, but really slow wave sleep is the energetically most conserved state that any mammal has available to them. And so if the whole point of hibernation using torpor is to save energy, then why wouldn't they, if they just had to be warm for biochemistry, spend the entire time sleeping? It makes perfect sense.
Oh, I agree with that, and it was shown in squirrels, so that's where still the I say the thing is not very clear, because in hamster that sleep after seems to be actually very true sleep. So I know it's still very mysterious to me how the two are connected, unless to this logical approach in which they are kind of, you could see non-REM sleep is a lesser parent than torpor, or the entry point for torpor. But other than that, maybe serve a very different evolutionary purpose.
Yeah, I think that's another thing that Craig Heller showed pretty definitively with nice experiments, that the entry into torpor starts from slow wave sleep.
Yes, I'm still a bit skeptical, but I couldn't see how else could happen. But yes, let's say that I agree with that.
So related, and another question is, does anybody know if there is a role that deep meditation has in reducing basal metabolism? Deep meditation being kind of a intermediate state, perhaps, between full awake and asleep.
Yeah, there's a book by Bob Elsner, who passed away recently, but that compares it gives a lot of stories about yogis and remarkable metabolic suppression. And I think there's been something published since then, but I wouldn't be able to find it. But I do believe I heard people talking about it. But it's extreme metabolic suppression.
Yeah, I'd heard stories about that, and I didn't know if they're just really one-offs, or if there's enough evidence that is controllable under some circumstances anyway.
So actually, Bob and Craig Heller tried decades ago to measure this in yogis, and they were not able to pull it off with the equipment that they had at the time. But that's what I think I remember hearing, is that it has been done more recently, and I can sure try to look for that. Maybe I can come across it and share that with you.
That would be great. That would be very interesting. Okay, so next one is, do hibernators show any consistency in their body positions during hibernation that might imply that they use gravity in some way as part of the hibernation process? Obviously, gravity being important if we're going to be having people in space. Anybody?
Our
I mean, the
ground
main,
squirrels,
go ahead.
our ground squirrels, I think most hibernators assume they sort of curl up. And the idea
fetal
is that
position, whatever that may be for all of them.
exactly, yeah. And the idea being that it minimizes exposed surface area, so it reduces the rate at which heat is lost. And then, which is different from when they sleep, where they're more splayed out, I think. I'm not sure, that's in our 13-line ground squirrels. I'm not sure what it's like in there, but from the ground squirrels, I don't know if, that's a really interesting question. And I don't know what, if any, role gravity may play in that.
I don't think it's required. I think it's part of the phenomenon because it's all about energy conservation. So they do everything they can to minimize heat loss, but I don't think it's required. So I don't think that gravity would be important. And that's a question, whether NASA would ever be interested in supporting a hibernating species in space to see if they hibernate.
Yeah.
That would definitely be interesting.
Yeah.
Dr. Story, back in the day when you were in the caves with the bears, did the bears do the fetal position thing too?
Oh yeah.
No. There was a project from JAX, the Japanese Space Agency, about taking animals into space and actually having them hibernate. So that idea has already been started to be looked at. It was a hilarious disaster. If by hilarious, you mean chipmunks just so angry they could beat you down. So it's really hard. You can upset a hibernator just by coming in and turning on the light for a second. They're not profound hibernators. You can rustle them. So no, I don't think hibernating in space starting it in space is a good idea.
Do you
I think there's
a species
some
one could use for that, conceivably?
Well,
there's some anecdotal or some belief that in some of the Russian, are they Bion, the autonomous spacecraft that had a bunch of animals in them, on those missions that certain animals, I think it's tortoises, were believed to, they survived the ballistic re-entry, partly, and I can't remember what happened on the mission, but everything else died, but the tortoises lived, and the belief was that they induced metabolic depression so as to reduce the rate of consumables use or what have you. But there were no measurements taken of metabolic rate. But
so who did this research, Dr. Reagan?
I think it was one of the Bion missions.
Huh, okay.
Yeah.
Russian. So I think it's a satellite.
Just Google Russian tortoises in space.
How many hits could you get, right?
Probably not a lot of hits, yeah.
No, there's 1.3 million results.
Oh, wow.
But I want to put out a question then, and maybe Matthew can say more about, wouldn't be torpor the perfect way to send actually animal in space? If we were interested in doing more research up there, or even one day send actual animal to the moon or to another planet, they can enter torpor. We know we could do it pharmacologically. So wouldn't that be easier than put an animal on a rocket that I'm sure is a little bit of a stressful event for whatever animal is there?
Yeah. Well, yeah, I mean, theoretically, it would reduce it would reduce the rate of oxygen that would need to be brought on. It would reduce the amount of water and food, CO2 scrubbing. It would definitely help. And then if there is some intrinsic radio protection that comes with it, then sending them to the moon, for example, outside the Van Allen belts where they're exposed to galactic cosmic rays and what have you, they would be protected from that, such that when they arrived at their destination, the moon, say, or something, that they wouldn't be, depending on the experiments, assuming the experiments wanted to start on the surface of the moon, say, or an orbit, lunar orbit, with animals that had suffered minimal radiation damage, then it's a potential way to do it. And I mean, mice, like, one wouldn't need to pharmacologically induce it in mice, for example, because you could just, make sure their environment, food, and ambient temperature were such that they would naturally enter those states for part of each day anyway. But I mean, I guess everything, you know, contributes. So if you could reduce their metabolic rate for 35% of each day or something by X number of percentage, and that's proportionally less you need to bring with you. And so I think there's some validity.
I think you could really store them like in the movie. I think if you're taking like a rabbit or something like that, or a mini pig, then the capsule would work for an animal, and then you get your mini pig animal farm on the moon. Or
I think a ground squirrel farm is the way to go.
Ground squirrel, okay.
Especially Arctic ground squirrel, because they're big. You can get some meat out of them.
Not to mention, it's pretty cold on the moon.
Yeah.
Very good. All right, so I'll move on to the next one. Are many of the, so not ask it quite that way. A number of the brain tissue RNA transcripts were found to vary very consistently across torpor cycles. Do any of those derive from genes shared by humans?
Almost all of them, but not all of them. Yeah, but pretty much all the genes we detected can be annotated against the human genome.
Do you see any intriguing hints from the ones that aren't included?
Well, that's really an interesting question, right? Because it takes a lot of time to figure it out when they're not already annotated, what they might be doing. And I know I personally have dug into this a little bit. We have genes that are transcribed, and some of them are quite strongly differentially expressed. Some of them are microRNA precursors, and they make novel microRNAs that are not identified in humans or mice where the annotations largely originate. But others look like protein coding genes that nobody's ever identified or talked about. And in at least one example of those, I can't even find the homologue in mouse. And the human homologue has been beaten up by insertion of transposable elements, making it look like it is it's a functional gene in this hibernator. It's induced in winter for hibernation, and it's been beaten up in non-hibernators to the point where it's not recognizable. Now, that's a really interesting gene to go further with. But this is like a one-by-one, time-intensive process to identify those and then figure out what they do. Right. So I'm sort of stuck.
And then get to the knock-in sorts of things to put it back in those where it's beaten up.
Yeah. Mm-hmm. I mean, what I'd like to see is that in other hibernators, distantly related hibernators from those other lineages, that gene is also conserved. But again, the genomes out there are not necessarily good enough to actually have a robust answer to that question.
Excellent, thank you. So this is a broad question, probably applies to everybody in one way or another, or at least the goal would be somewhat of a consensus answer. So the workshop was described as having done a good job of reviewing the topic in general. However, could the panel enumerate the major mechanisms or hypothesized mechanisms for torpor versus those that are deemed more minor that were brought up? So what are the sort of the major top 2, top 3 mechanisms that you think are involved? To me, it's also a little unsettled in my own head as to kind of what comes first. What's the chicken and what's the egg here, in some sense, as to what initiates, what may be driving animals into torpor or back out. I very much like the model of the buildup of some biomarker that decays over time, and that pops them back awake, and then they generate more of it, they go back to sleep. And
adenosine, you got to remember adenosine.
I'm sure maybe we'll have 6 answers to this one.
That's my answer.
I figured yours was going to be adenosine related. So do you think, more specifically, do you think it's a endogenously driven buildup of adenosine that is either circa annually or whatever, or external temperature driven? How does that, where does it start?
And I like to imagine it's a positive feedback, feed-forward cycle that adenosine is regulating the epigenetics, which is regulating adenosine kinase expression or tanocyte cell proliferation, although that is then promoting it. But that's clearly a gap.
Fair enough. Other answers.
I know, I don't know. I mean, I can say there's a lot of changes that are associated with the different physiological parameters that we can measure, but I don't know who's driving and who's following. And I think that's a challenge for the future to understand that.
We're okay with speculation here too. So I understand this is a scientific forum and we're always afraid of speculating too far, but we're open to taking those sorts of risks, Trish. Matteo, I think you're muted somewhere.
Neuropeptides.
Any other answers? Or anyone else want to take a stab at it? We're going to have just one answer of adenosine because it really seems like somebody else is probably got a favorite horse in this race.
Okay, Kelly, I have a question for you. You liked glutamate for a while. What about that?
So we thought that was driving that was the cue for arousal to normalize the glutamine balance, but the recent studies have not supported it. So those are some papers that'll be coming out soon. So yeah, I don't know. And even the nitrogen, whether it's through the glutamine or not, just Sarah looked at just infusing ammonium acetate to induce arousal. And it did induce arousal, but in a very odd way, a very, very different pattern, timing of arousal. So that's still out. But I still like to imagine that intrabout arousal is to normalize, metabolites, and that there is a metabolic cue that promotes that. And I still think nitrogen is a possibility, but we don't have a clear answer on that at this point.
Yeah. I mean, I guess the way I see, for anybody who wants to put humans in space, I think intrabout arousal is a problem and you don't really want it, right? But we need to understand why they do it. Why would they waste all that energy? And their whole adaptation is for the point of saving energy. And maybe it's a clue to understand the mechanism of inducing torpor. And so that's why I'm enthusiastic about these time points where we consider what happens as they cycle through those phases of entrance and arousal again, because it makes no sense that they do this, really. There must be something going on that's really important, and it could provide the clue for what we're looking for. But we haven't found it yet. I don't believe anybody knows, not even adenosine.
Isn't it estimates up to 90% of all the energy used by hibernators throughout hibernation goes to fueling these arousals?
Yeah,
so they're as important as hell. But
yeah.
It seems like crazy to me, like they wouldn't, being an absolute know nothing in the field, seems to me like it's something they can't avoid for some reason.
Right, but if there is a metabolite that's essential for torpor and it's depleted, so they have to wake up to make more because they can't run the biochemistry at low temperature. I mean, that makes perfect sense. And we should be able to figure out what it is by studying these time points.
Right. And a window into that could be the lemurs that you talked about, Sandy, which the individuals, depending on their ambient temperature, they will or they will not induce these arousals, which, and it's believed to be a temperature dependent thing. So that's
if we could just put ground squirrels into those conditions, and if they did the same thing, if they could ride ambient temperature passively like a lemur does, I mean, then we would have enough animals to do the experiment to figure out what it is, right?
I see. Maybe I missed that somewhere along, but so lemurs will actually not won't awake up if it's cold enough?
Actually, I have the slides. If you want, and you'll let me look, I can show them to everybody.
It's actually the opposite, though, Gary. It's they won't arouse if their body temperature is sufficiently high while they're in torpor.
Oh, interesting.
Which is consistent with bears too, that don't get so cold and they don't do the interbout arousal.
Yeah, and it's around the same temperature too, isn't it? Like right around 31 or something?
Yeah,
right around
30. Okay, so
Yeah.
let's
Any
see. If I
hypothesis for
have,
why that happens?
do I have permission to share my screen?
Absolutely.
Okay, let's see if I can do it.
Yep.
Okay, so let's see if I can get it here. So this is not my work. It's from Dousman and Heldmeyer from a number of years ago now, but they had these beautiful studies. And I'm sorry, I don't know how to make all these. Let's see. Okay, so let me get to the right beginning of it. So there are 3 temperatures being recorded here. The blue is the body temperature of the lemur. The red is ambient temperature outside, like on the ground. And black is the tree hole where the lemur is undergoing its hibernation. And what you can see is that the tree hole stays pretty cold. It's right around 20 degrees continuously for this whole trace. The ambient temperature is swinging up and down, but the body temperature of that lemur is spending, say, 5 days in torpor and then spontaneously rewarming, just like the ground squirrels. So 5 to 7 days rewarms. But that same animal can get up and walk to a different tree hole sometime during hibernation. And here what you see is that tree hole doesn't ever get above 30 degrees. Okay, so red again is ambient outside the tree hole, black is in the tree hole where the animal is. And then you see it kick up its metabolic rate every few days, again, just like it did before when it was colder, to get that body temperature over 30. But the same animal gets up and walks over to another tree hole where ambient temperature is cycling higher than 30, and now it is passively riding that temperature curve of the ambient temperature. So that tree hole is so poorly insulated, it's basically the outside temperature. And the same animal can stay torpid for weeks. So this is a pretty profound and interesting behavior. And it suggests that there's something about the low temperature that maybe it's a metabolite that can't be gotten rid of or made that needs to be there in order to have torpor. It looks like maybe something gets depleted and they warm up and make more. But you would get a handle from studying an animal with behavior like this on what it is that's so important, right? Because whatever it is can't be fixed here, but all you do is warm up a little tiny bit, you fix it, or you passively take care of it completely as long as your body temperature goes above 30. And nobody's ever put a ground conditions like this. We don't know. Maybe they do this too. We just don't know. I think it would be worth knowing because, I mean, you just can't take enough samples from all over the body of this little guy to figure out whatever this magic stuff is.
Indeed, that's very interesting, and yeah, a lot of hypotheses that could potentially explain that.
Right.
More gaps to add to our pile.
Right.
Yeah.
I'm going to move on to the next question here. Has there been anybody know of any recent work or interest in the rate of infections and disease progression during hibernation? It may be organism dependent, but any input or insight people have?
Again, thinking mostly of older literature that's been around for a long time, most pathogens that live in animals that are supposed to be at 37 degrees Celsius are also suppressed. Their growth and their pathogenicity is suppressed by the low temperature of torpor as well. And what we know is that when ground squirrels rewarm, their immune system activates again, but it's completely suppressed also in the cold. So it's the host parasite battle continues during those interbout arousals, but it's generally suppressed by the low temperature when they're torpid.
Great, thank you.
What about in white nose syndrome? Because isn't that a cold loving?
Yeah, that's another,
fungus?
that's a fungus. So I'm thinking mainly of bacterial or virus infections.
Okay.
Is that more active or at least remains active when cold? Does anybody know?
I believe it does. And
They're susceptible during hibernation and it gets really out of control.
Yeah.
So we'll have to make sure not to have any white nose pathogens on board before we leave.
Yeah. You guys are pretty self-conscious about
They do
pathogens
pretty,
on board
yeah,
of
they do a
any
pretty
kind,
good
right?
job scrubbing.
Yeah.
So Dr. Strongman, probably we'll have time for one or perhaps two more questions before we start wrapping up the session.
Let's see. So here, another question here is, does anyone see possible connection between miRNA, exosomes, and virus particles or virons, as psychiatrist Dr. Andrew Kaufman from Philadelphia has suggested? If nobody's familiar, then we'll just skip that one. I think we'll skip
You want to
that
keep
one.
going?
Yeah, no,
I
that's
mean,
fine.
exosomes package microRNAs and move them through the body. I think that's pretty well established from non-hibernators. Gosh, there's a protein that's a gag-derived protein that it's used in the brain, and it moves things from cell to cell. That gag-related protein that's been taken up and now part of our genomes is actually one of the hibernation-induced proteins, but sorry, I can't remember its name. It's one of my 5,000 differentially expressed proteins.
Which you don't have all memorized. I'm shocked.
I hope that the paper will be released soon and everybody can explore that data set because it's far too vast for me to interpret.
I think this will be the last one. So we talked a bit about muscle and bone issues while hibernating. What about deconditioning during hibernation? Does this happen, and to what extent? Or do they just wake up and they're ready to run? It seems like at least the squirrels might be ready to run if they start mating as soon as they wake up. And does anybody have any idea how on earth that's possible? Because I know I couldn't do that.
Well, I think they start mating when they emerge from their hibernacula, but I'm not sure that they emerge from their hibernacula right as they arouse from like their final arousal out of torpor. In fact, I think in hibernators in their natural environments anyway, as the hibernation season progresses towards its end, the rate of arousal starts to become greater. So they arouse at a higher rate, more regularly. And I think the depth at which they depress metabolic rate when they go back into torpor is not as deep as it is in the middle of the hibernation season. Is that right?
I think that data you're thinking of is free ranging, and that's because the ambient temperature is not as cold. But the Arctic ground squirrels cache food and stay underground for about a month before they come up and they're ready to mate.
Is that only males? Because that's my understanding. Is
yes.
The males are the cachers, and they spend time warm, growing
underground.
Their so they're ready to go the minute they come out. Yeah.
That's what I remember.
Okay, well, thank you very much, Dr. Strongman, for reading the questions and making sure that we were catching up with all the questions. I believe we received close to 110, 120 questions, so that was a really, really good engagement from our attendees. I think this was a very, very successful workshop. We really appreciate all of you participating today. I think that we have a really good understanding now on what's the state of the field. We have a good understanding now on what are some potential gaps, some potential areas to explore. And thank you very much. Really, really appreciate it. Dr. Donovan, I don't know if you have any last words for the group.
No, I really enjoyed this. We have so many memes, especially from Dr. Stories. We wrote down all the memes and we'll be tweeting all those. But no, I really appreciate all of you taking the time. And I think my favorite part of the whole thing was just to hear you guys talk among yourselves. Sometimes people just enjoy being a fly on the wall when you guys are the experts. So thank you, and we'll be taking your ideas forward. And please, if anybody in the audience wants to send us their ideas via email, etc., we'll be thinking about this for solicitation topics in the future. So there's opportunities here for everybody.
Thank you very much, everybody. Please stay safe. Have a nice weekend, and let's stay in touch. Thank you very much, everybody, and thank you for all our attendees for staying today. Thank you.
Thank you
Thank
for a
you.
great
Thank you, everybody.
Thank you.
Thanks for having us.
Take care.